You had to bring on all that scientific expertise. And then as you formed up the organization, what did you have to do from a personnel standpoint to support the development of this science?
Yeah, this was really challenging because when I took over, the company was geared to run clinical trials. Mm-hmm. It was being set up to enter later stage development. So there was a relatively substantial CMC team and the clinical development and regulatory team. So the bulk of the organization was CMC and clinical. A lot of talented individuals. We built a super team over the years. And then we had very little science. The labs had been shut down earlier because when you're in clinical development, you have less need for that, and the clinical data analysis was outsourced. So we only had 1 or 2 scientists really, and the CMC and clinical team. And then obviously that's not what you need when you're building a preclinical research organization. So
it's important though. It is important.
We sort of built down one part of the company and built up another part. So the clinical development and CMC group is down to just 1 person in each.
Wow.
That's it. So that's been tough, but everyone, I have a very sort of open dialogue and never hide anything and tell people the truth. And I think people understood the rationale, and this is what has to be done, and it's just the way things are. So there's never been any grudges, or it's sad, but that's just the way it is. And then in parallel, we've built up a new science team in Stockholm. So we've both been downsizing and recruiting at the same time.
Yeah, yeah, balancing the demand, right?
Mm-hmm.
Yes. So we've added scientists, and we have 1 more now, and actually an American lady coming in after summer, joining us in Stockholm to manage the lab there. Awesome. We're just moving into a new lab, and so that side is growing. And in Oslo, we're down to it's just me and finance, two finance people and myself. So we're just 3 people left in Oslo. Managing listed the company, so it becomes pretty complex, and then the rest of the team is scattered with the bulk in Stockholm.
Yeah, circular RNA being buzzy as it is, at the same time, there's it's a small, probably but powerful group of companies that are working in development of this space right now. Does that create competition for talent? Like, is it tough to find people who can come in with their feet on the ground?
There.
I think everyone in my team has been approached by the other circular RNA.
Yeah.
It's a young field. So and there was a lot of skepticism generally towards that original paper we published also. It took a long time to get that published. I think it's not until 2013, so about 10 years ago, where it became obvious to everyone that this is a real biologically relevant and functional class of non-coding RNA. Mm-hmm. And then from 2013 onwards, it's started growing. Initially, it was kind of an academic corner of people, a few people working on this, until Alex Vesseloff out of the Dan Anderson lab, which became the foundation for RNA, showed that you could use it to express protein in mouse. And then from there, it kind of exploded a bit. But the point I'm getting at is that it's only 10 years since it started. And I think for the first 5 of those, very few people worked on it. So it's extremely hard to find any experts. Yeah. There are very few scientists around who understand the biology of circular RNA and come with experience in the space. So that's maybe what's holding back most the development. Is just we're still playing catch up in building the biological understanding and also learning the scientific techniques, the manufacturing technology, and having enough people who are knowledgeable about it.
Yeah.
Yeah. I'll ask a question that'll tee you up, and get back into the science, because there's the historical biological foundation of information here that's hard to find, and then you winnow that down further when you talk about your differentiated approach to the space. Obviously there can't be, even in a small but hot industry, there can't be 3 or 4 or 5 people doing the exact same thing. So you winnow that down. Now you've got to take people who know what they're doing in general circular RNA, and then you've got to teach the platform, you've got to teach the differentiator. So let's talk about that a little bit, and that'll tee you up, Anna Rose, to ask some more questions about what Circio is doing.
We'll find out.
But is it the in situ production approach at Circio that is sort of the cornerstone of the differentiator there, or is there more to that story?
Yeah, absolutely. So when we got into this, we see that all the other circRNA companies are more or less doing the same thing. In vitro transcribed circRNA as LNP package. This, in many ways, repeating the BioNTech and Moderna play, just using the circular format. And once you figure out that manufacturing, it's a sensible thing to do because it's likely to be cheaper and better. So I think that's what everyone jumped on first. And still, when I talk to even people I've had 2 or 3 meetings with and explain our technology, they still think we do that.
I was just going to ask, is that confusing to people?
But remember, so first of all, we came from the virus angle. We were a virus company. And then secondly, we look at this space and we say, yeah, that's obvious. Circular RNA is likely to outcompete mRNA, has these chemical advantages, but these also apply in other contexts, and they've so far been completely overlooked. No one had just made that simple effort of making an expression system using circRNA instead of mRNA from a vector. And we saw it as an opportunity, a virgin territory in many ways. So and we're a small company. We're based in the Nordics. We're much less capitalized than these other companies. Why go head to head against them when you can sort of have this niche that no one's taken, and rather be the big player in another area of circular RNA. So we don't view these other companies like Orna and Sail and now Orbital as well as competitors. This is complementary technology. We are simply using circular RNA advantages for different purposes. And I'm only aware of 1 or 2 other companies and groups really working on the same approach as we are doing.
Okay. I was going to say, when I was first introduced to your company, my brain sort of exploded when I realized what you were doing, right? Because again, I think in the RNA space, we're always talking about IVT, right? And so to go to your site and to see the words circular RNA and also AAV, right? Viral vectors, it was kind of, it gave pause, right? I was like, what is this? How does this work? So, I think that it is a really interesting and competitive angle, but I also love this concept because I think in the RNA space in general, we're all talking about how we can create a pretty well-rounded toolbox, right? It's not just one modality, one method of development. I think having this version as well could be a really interesting addition to that.
We're bringing the advantages of circular into novel areas. So a synthetic IVT circRNA, I don't think will ever be relevant in a gene therapy context because the durability isn't good enough. This is something it's better than mRNA, but still it expresses for 7 to 10 days.
Yeah.
When you're talking gene therapy, you need something that can express a payload for years, at least months. So we're just solving different problems here, and I think we are enabling circular RNA to be utilized for new things.
I was going to say one of the things too that I just kind of wanted to return to really quickly is one of the biggest challenges you'd mentioned is just that lack of sort of foundational research in this space because it is still so new. I'm curious, as you guys are working to sort of develop these products and position your science, where are some of the biggest groundbreaking foundational research elements to circRNA that we need to see, or that you're hoping to make as well to help, I'd say, kind of heighten the way that you're using circular RNA at Circio?
I still think the delivery is what holds vector technology, and delivery is what holds back the potential of a lot of therapeutic areas still. So everyone wants to get to a point in the future where we have repeat dosable nucleic acid therapeutics that can traffic to the specific tissue or even cell type that you want it to. You give it IV, it goes where you want it to. It will express the gene there for a long period of time.
Only there, right? And only in that specific spot.
Exactly. And the underlying molecular biology exists. but the delivery doesn't exist yet. We simply don't have that kind of technology yet. I think you look into the future, we will get there. But right now, we're still dealing with relatively crude things like an LNP that you just rely on passive trafficking to the liver. So that means you can deliver stuff to the liver. So obviously that's where you start. First gene therapies, you pick diseases where like either muscle or liver, which you can easily access. But the moment you start being able to get these into brain, heart, other areas, you can start dealing with other diseases. And inevitably that's where we're going to get to in the future. But we can't solve everything. I think you need to focus right on what you're an expert in. Some companies are vector companies. They make the best vectors like AAV engineering and having the AAV go where you want it to. Others are delivery companies making chemical compositions that enable packaging and targeting of molecules and therapeutics that way. And we're the sort of molecular biology. We create genetic constructs. And we are relying on partnerships to solve these other challenges.
I was going to say, you have been meeting with quite a few partners in the past year or two, right? You've been trying to shop your technology around and meet somebody who's complementary. So yes. I'd be curious if you could share a bit more about how you're approaching partnerships today and in the circRNA space, in that AAV realm. And what are some of the most important questions that you've been asking your potential partners?
Wasn't you asking a partnership?
I know. Look at that.
Getting off the scientific context.
Nice.
What I'm most excited about for the future is DNA-based gene therapy. So by that I mean synthetic DNA format vectors that are redosable. And I think that's where gene therapy will get to in the future. We're going to probably move away from viruses because of more difficult to manufacture. You get immune response. You can't really redose them, at least not as it is now. But if you could deliver a synthetic DNA construct efficiently, that should be much more durable than an RNA-based therapeutic, and also it should be redosable. So that's sort of our long-term goal, to make DNA format gene therapies expressing the gene that we're delivering from our circular RNA circVec technology. So in terms of partnering, that's of course something we're very interested in, meeting up and collaborating with DNA vector companies that have these technologies are starting to develop them. None of these have moved into the clinic yet, so this is all kind of early stuff. But that's a high focus for us in parallel with other delivery technologies like I described before. And this is maybe more kind of the long-term goal. Of course, we need to keep in mind, like I said before, what's our lead therapeutic candidate, and there we're still prioritizing AAV because it's something we believe that we can fix fast. It's much more straightforward, and you don't need all these other moving parts to work together. You can sort of make a simple switch to an AAV and have it work. So there we're looking to partner with any AAV company. Really, I think once we show this works, and we can improve on the protein output from an AAV, every single AAV company should be interested in this technology. Why would you want to make an AAV expressing for mRNA if you can do it via circRNA and get more bang for your buck? Mm-hmm. So there, I think we're trying to make ourselves attractive for AAV companies, and we're looking for collaborations in that space, and of course in parallel advancing our own AAVs.
Mm-hmm. Do you have any, let me put the question this way. What can you share with us in terms of what a Circio pipeline might look like?
So the lead program we are working on right now is in a disease called alpha-1 antitrypsin deficiency, AATD. It's a disease that gives symptoms similar to COPD. You get breathing issues, and also it has a liver aspect to it. And it's a unique genetic disease because you have a mutation, and that mutation results in a misfolded protein. And that misfolded protein, one, it accumulates in the liver. So over time, you get liver toxicity, and it can be very serious. Patients need a transplant in the end. And then the lack of the functional protein causes these problems in the lung, like the COPD-like symptoms. So it's a unique genetic disease where you need to deal with two problems. You need to both remove the toxic form of the mutant protein, and you need to replace the functional one. Because we use circular RNA in our expression, we can do this trick where we're able to knock down in the patient the mutant form and replace from circular RNA the functional variant. And it's only a single nucleotide difference between the two. So these are extremely similar. But because we use a circular RNA for the replacement of the functional protein, and naturally in the body there will be an mRNA making the mutant form in the patient, we can distinguish the two very cleanly, knock down one, replace the other. So I think that's where we established a unique selling point, USP, and with our CircVec we can do then what we call remove and replace. So we can have dual functional gene therapy where we shut something off at the same time as replacing the wild type. Yeah. And therefore we purposely looked for these type of diseases where you need to solve both of these problems. And that would be really hard to do with any classic mainstream AAV or gene therapy. The only other technology that could deal with this in a similar way would be gene editing, which of course is exciting, but also very early. Mm-hmm. So in that regard, maybe gene editing is our real competitor, not another circRNA company.
Yeah. What is
long story, but remove and replace gene therapy for AATD and beyond. That's where we're heading.
What does the landscape look like for other indications where remove and replace, where that approach would make sense?
So when we started our screen for diseases, we thought actually there were many more, but most genetic diseases, you usually have one problem or the other. Yeah. It's quite few where you have this dual issue, but there are a couple where it's cystic fibrosis is another one. Where you both want to remove and replace at the same time, and we're looking at several other candidates that we haven't disclosed yet.
Someday soon, hopefully.
Someday. You don't necessarily need to. You could also use this knockdown capability to augment other pathways. So it doesn't necessarily have to be the exact. You're shutting down and replacing the gene. You could pick other targets as well, where you're able to modulate pathways to boost the potency of your therapeutic.
Yeah. We're running a little bit tight on time here, Anna Rose, but I don't want to cut you. If you've got other burning questions regarding the RNA landscape and the science going on there, now's your chance.
Oh man, that's a lot of pressure.
I'm sure you have more.
So much pressure. Go ahead, Matt. I'll cede the microphone this time.
This time, yeah. A rare occasion. Yeah. Excuse me. What's the next? I mean, you've mentioned there's a lot on the agenda, right? You've mentioned there's a lot going on there. In terms of what Erik's going to do next, like what's the next immediate challenge that you, as the leader of Circio, are compelled to wrestle to the ground?
So you can only pick one.
Yeah, you can only pick one.
Only pick one.
Also, I think the challenge is to balance this need for having a lead, focusing on a lead candidate at the same time as expanding and building your platform.
Yeah,
and we simply don't know yet. When do we reach the maximum potential? Where does it work the best? And we may discover that our platform, it may have an even better application. It may be more powerful here. Maybe we should use it with this type of vector. Mm-hmm. So I think it's important to balance here sort of the platform versus the product candidates. And like we pivoted before, I think we need to be ready to pivot in the future. If we come across something that looks more promising, we need to be ready to switch, because we still don't fully understand the potential and the best applications. We have a pretty good idea, I would say. We know which direction we're going in, but we're certainly not married to that and ready to switch when the time is or when the data calls for it.
It's a perfect setup, perfect segue
Couldn't have been more perfect.
for my last question for you. I mean, as I said from the outset, if you're a biotech leader and you haven't pivoted yet, like there's gonna come a point where on something you've got to make some important decisions that could fundamentally shift what you're currently working on. So having lived through this and being that guy who does it optimistically and proactively, embraces
He lives it too, right?
Yeah.
Let's be honest, he's lived it professionally and personally.
For sure. I mean, in some people, I think it's just inherent, like it's a natural embrace. It feels good even. For others, not so much. So what advice would you share to other biotech builders who, maybe struggle with it or maybe haven't seen it yet?
I think to some degree, this is kind of a personality trait. I'm also the type of person on a holiday, I like to change plans and go to a different location. Let's do this instead. Or
Yeah.
I don't mind the change of plans or something unexpected happening. I find energizing. More sort of being too focused for too long can get boring. But some people are more like that, and then maybe biotech is less an ideal organization or industry to be in. I think you need to kind of love this fast-paced, dynamic aspect, see new opportunities when stuff inevitably fails. And other things work and stuff, unexpected things happen, and you need to be able to quickly adapt to that. And let it guide you, and don't roll over and die.
Don't roll over and die.
Not to put
the subtitle for this.
not to put too fine a point on it.
It's what makes it fun. But you can't be the nervous type. Clearly, if you're after long-term predictability and stability, then biotech is going to be a stressful arena for you.
Have you thought of photography or meal kit making? There are other options out there for you.
No, that was certainly there were pivots there as well.
I can imagine.
Yeah. Well, this has been a fun conversation, Erik. I've appreciated every word of it, and hopefully we didn't do too much bouncing around Anna Rose. I mean, I think we
restrained ourselves.
Yeah,
Really.
yeah. There's so much more we could talk about, but we'll let you off the hook for now. We'll let you get on with your day. But I appreciate you coming on and spending some time with us.
Yes, thank you.
Likewise, thanks for a great discussion.
We'll do it again sometime.